introduce it. The intention here was to basically come up with the technical background. I'm going to actually explain in some detail exactly what CRISPR is, because CRISPR is sort of the scary word, I think, that helps to push a little bit of the concerns about this. And this is I think on Monday, this is the quote that came to my email from several of the news outlets. As a result, this program may be widely perceived as an effort to develop biological agents for hostile purposes and their means of delivery, which, if true, would constitute a breach of the Biological Weapons Convention. So if you get that in your email, you're probably going to have a bad day or a bad week, particularly when it's in reference to the particular program that we're working with here at DARPA. You guys are coming in from it from a perspective of where you are the consumer, shall we say, of the news. Just as a reference, some of you may not be aware, Science is a relatively well-known journal, sort of like one of the most well-known journals out there, though it's really a historical magazine. So it tends to be short, tends to be a little different than some other journals for reasons. I throw this out here because this sort of gives, like the always, we learn by pictures, even in science. And this is a concept that they were outlining. They're basically talking about horizontal gene transfer, which they like that word that they sort of coined here. But facilitated, basically, horizontal gene transfer. We talked a lot about the idea of DNA does actually move between organisms horizontally, meaning like between organisms that are not doing that through sexual means. Just in some cases, even, can anyone remember what was the one that's the easiest to do this horizontal gene transfer? Anyone?
Cyanobacteria.
Cyanobacteria. They're naturally competent. They'll literally just suck up DNA from the environment and stick it randomly and try it out and see how it works. So horizontal gene transfer varies depending on the organisms you're working with. And you can kind of imagine that there's a lot of reason that there's not horizontal gene transfer. You don't necessarily want to just pick up DNA and try it out and see how it goes. Generally speaking, it does have function. That's the benefit. The downside is, of course, things like viruses and so forth may not have your best interests in play. The key element here is they do focus a lot in CRISPR, and a lot of what I'm going to talk about here is actually going to talk about CRISPR as well. And the scenario that they paint here is that the insect's essentially delivering to a CRISPR target using CRISPR. And most of you have at least heard it. This is the so-called gene editing capability. And if you were to try to target, for instance, plants' ability to reproduce, seed production, ultimately you could essentially create a food shortage via this type of a vehicle, which you can kind of imagine if you're wanted an offensive weapon. Hungry, starving soldiers may not be what is the vision, I suppose, here. There's a lot of aspects of this that I'll be talking about, and in particular, there's a tremendous focus on CRISPR, so the next few slides are dedicated to really understanding what is CRISPR. You may or may not, any of you have that in the intro biomolecular class. Good bit? Okay. With that in mind, okay, sort of here's the CRISPR system itself is actually an antiviral mechanism for bacteria. Something they didn't sort of understand, but they knew that bacteria had this amazing ability of once they saw a virus, next generation or so, they would be resistant to that virus. They weren't exactly sure how that was happening. What it represents here is there are elements here. This is sort of a working business in here. And then these elements here are the repeated elements that are the bad guys. These represents, if you want to think of it, as virus A, virus B, so on and so forth. And they saw these essentially real short tandem repeated DNA sequences that didn't seem to be doing anything. But what they're actually doing, if you take a look at how this works, the Cas9 is a big enzyme. Okay, we'll talk about the bigness of that in a minute. The tracer RNA is a means by which that element can go over to here and take that identification code, if you will, for the bacteria and create this element right here. That element basically gives them the ability to say, I want to destroy anything that has that green sequence. And you can kind of imagine the value of that is that once you're exposed to something bad, if you save a little piece of it, and that piece is unique, i.e. not in yourself, okay, then anytime I see that DNA, I would destroy it, and I could protect myself against being infected again by that virus the next round. Okay. The important aspect of this is that in prokaryotes, which I emphasize up in the top there, is that it is designed to destroy DNA. Okay. It is designed to destroy viruses, and it will destroy also bacteria as well. That's why they have to pick a unique sequence that's not in their own genome, because if they did, then the CRISPR system would actually do DNA breaks as well in their own. And so that functionality is inside this enzyme. Keep reminding that the essentially the Cas9 is the enzyme responsible for making the actual cleavage cut there. There's a lot of nifty details. It's called the PAM recognition site and so forth. So it doesn't actually, what it does is it has the recognition is here, and then it cuts over. That's actually really useful from a standpoint of allowing it to do cuts that are not based like a restriction enzyme on a specific site, but a certain distance away from a recognition site, and that's very useful in terms of its ultimate use. The next one then is to see that this works quite a bit differently in eukaryotes. This is the important part. Okay. In the eukaryotic systems, okay, the bacteria, if it sees that sequence, it'll actually break it, and it will be lethal. Eukaryotes are actually pretty good at repairing DNA breaks. And as a result of that ability to actually repair the DNA breaks, what you end up with is, in this particular rendition of it, okay, this is again the Cas9, that's the enzyme. And here, this is the recognition piece. And they figured out pretty quickly that they could make a synthetic form here. So rather than having that piece, we go find the other piece and make a piece of that extra one, I can just make it all as one piece of RNA. They refer to this now as the guide RNA. And so that guide RNA then will actually give rise to recognition capability here. Okay? And you can actually then, in just one transcript, if you will, create what you need here to guide this thing for the actual cuts. Now, remember now that eukaryotes, when the cut occurs, have this ability to do repair. In the process of doing the repair, if they repair without putting necessarily something in there, if they get it out of the reading frame of the DNA, it's functionally, you can basically mutate and destroy that specific gene you're after. That's why we refer to those as knockouts. So one of the easiest things to do with CRISPR in the world of the eukaryotes is actually to do a targeted knockout of a gene. If you also happen to have an insertion sequence that is present during the repair, the insertion then can find its way into there as well. So that's a knock-in. The knock-ins and knockouts are both done that. Plus, remember, this aspect of DNA insertion during repair has been around forever. It's how horizontal gene transfer happens in eukaryotes. Okay? But you can make it happen in a specific place. That's why it's been given the terminology gene editing. It's not just simply the ability to get DNA in there. That's not inherently hard. It's the ability to put it in a very specific spot. Yeah, if you've heard of things maybe like the gene gun, for example, and I mean, it's a pretty brute force technique. You basically shoot DNA in. And if that DNA happens to be in and around where there's some DNA breaks, the actual repair mechanism can integrate those, but you don't know where that's going to be. Even things like agrobacterium, which I'm going to mention in a minute, that have come up with mechanisms of doing that, they don't do it in a predictable spot. They just randomly put it into there. It's semi-randomly. It's where it's actively being, things are going on when it does that. So at any rate, this is now so you have an idea of the tool that they're, the scary tool. And it is a powerful tool. And you've heard about it in terms of gene therapy, so on and so forth. And in fact, that is the terminology that I've been sort of looking at it. But there's an interesting aspect of gene therapy where a lot of people think that gene therapy represents a genetic modification into the chromosome. And I'm going to emphasize in a little bit here that that's not the case either. Most gene therapies are actually not in the chromosome. It's actually based on a virus that's just swimming around. So with that in mind, then I just wanted to sort of, re-articulate more specifically the scenario that was outlined in the science paper. The reason they knew this is that this was and they call it a BAA, but it's a broad agency announcement. It's basically an announcement to the academic world, or the world in general, shall we say, that we would like a technology and you come up with the ideas. But we'll tell you what its performance has to be, very specifically. And so it's actually in various phases. So phase 1, you have to be able to do this. Phase 2, this. Phase 3, and it grows with time. Initially, one gene being transferred of any gene type to one plant. Next one, groups of that. Final phase of this particular project was to get the whole way of a actually fully contained system, but it included target plants like that the insect might like better, doesn't like at all, parasites that would actually kill the insect that's trying to deliver the DNA, trying to come up with a simulated environmental system in which you would actually have the ability to do that. So the starting point of that is, of course, a virus. And the virus then is the element that would actually be delivering then that. And so you can imagine that this will require putting in their mind, remember, this is a scenario in science, all of the elements for gene editing. I have to put the CRISPR in there. I've got the Cas9. I'm going to have to put the guide RNA in there. And if I'm going to put any type of functional gene, I'm going to have to put that in there as well. So the virus is going to have to carry all the pieces to the actual plant in some form or another. And I just took a picture off the internet here of a big virus. And the reason why is it's a big job that needs to be done. Next thing you need is a vector. Okay? In genetic engineering world, vectors are plasmids. In this case, we're talking vector as in a bug. And so there have to be some way in which that those bugs are going to actually take up the virus. So you have to make the virus, and then you're going to have to figure out how to get it. And for a variety of reasons, the very top thing here, all of these are phloem-sucking insects. They actually are, they go through and they actually have very specifically going after where the sugar is. So when you have the actual photosynthesis of the plant and it's dumping the sugar into the vascular system, I mean, it's the candy, shall we say, in there. And as a result of that, it's a precision delivery of these viruses. They are transmitted in the vascular system. It's not like you're just kind of going after the thing and beating on it. And as a result of that, it's an important aspect of some of the things we'll talk about in a minute about biocontainment. So at this point, you have the virus, the functions for genetic modification, the beneficial gene that you're trying to deliver, all assembled. It all goes into the virus. Now, or into the insect. I won't go into the details of that here because it depends on the system, but this is very specific. This is not a dirty syringe. The insect is not just simply going in, sucking on something, going to another one, dirty syringe going on. It turns out that the encapsulated virus is there actually has to go into the bloodstream, from the bloodstream, go into the salivary glands, and then from there, that's how it actually works. So it's not necessarily, in our case at least, our virus is not. But in general, this is the concept, and then of course the last part of this is how you go about then dispersing the insects in the field. And there's issues here, of course, you want the insects to die after they've actually delivered the helpful genes and so forth. And that's the elements that you can kind of imagine. And that's the scenario that, well, it looks somewhat scary, I suppose. The requirements from the science perspective here, the science CRISPR scenario, is that you are going to have to deliver the virus to the plant, systemic movement of the virus in the plant as well. The expression of the editing genes, remember Cas9 is an enzyme, the expression of the guide RNA to facilitate that, and then it's a successful high-efficiency editing of the mature tissue in the plant itself. All that's going to have to happen as they were trying to envision what was going on here. And it was outlined pretty well in the announcement that that's what their sort of vision was. It's important to understand here is this is not a single gene rare event transformation. We're not talking about getting one of the cells to transform here. We're talking about trying to transform the majority, if not all, the cells. And for the scenario that they were envisioning in that figure I saw, you would have to actually figure out how to do the editing that would end up inside the seed as well. For it to actually. So that's the scenario that we put here. So this leads to a need to actually understand transformation. And what was clear in many of the questions that I was being asked by the news outlets, that the concept of transformation is not very well understood. Because there are basically two forms of transformation here. Usually when we hear the word transformation, we tend to think of that as the scenario of when a gene ends up stably in the plant world into a plant. When we talk about in the bacterial world, transformation is when I take a plasmid and that plasmid gets into the bacteria. It's not in the chromosomes. So the important thing to recognize here is there's the DNA that you're actually delivering up here, and then there's the chromosomes. And in the eukaryotic world, the chromosomes inside the nucleus. Transient expression, all you need to achieve here is for that DNA basically to go inside the nucleus. Once it's in there, okay, then the machinery can take over at that point and do the transcription, translation, and so forth. But it does not have to go into the chromosome. And this is a very frequent technology. It's used a lot in the plant world because it's actually quite hard to get the other type of transformation where that gene, YFG is your favorite gene, okay, actually makes its way into the chromosome, okay? The key thing about this now, so this is stable transformation where now if that one cell that's been transformed figures out how to grow up to be a plant, okay? The nature of that is actually through techniques, several ways. Of course, one is through seed. If that seed can somehow make its way to eventually become the seed, then I will have successfully created a transgenic plant. Or if I can actually make that undergo somatic embryogenesis. And somatic embryogenesis is that thing where I say, yeah, that every cell in your hand is like a fertilized egg. It goes from, you have the egg, the sperm, you go to haploid, they come together, they become diploid, and then they divide a lot. And then they forget that they're a fertilized egg and they become your hand. Now the question is, can you convince the cell in your hand to actually go back to being an egg? And the point is that can be done in the plant world. So somatic embryogenesis is a process that I can make that individual one, and that's the important thing here. Is the specific cell in which the DNA has been integrated into the chromosome must now be the one that becomes the plant. If you don't do that, all you've managed to do is for that generation, you have a leaf that may have a couple of cells in there with the DNA and the chromosome, but it's not going anywhere at that point. When the leaf dies, it's not the seeds themselves. Okay, so this leads to then a couple of other things that I think are important to understand, because in the article itself you saw they were kind of saying, well, why don't you just spray them? Okay. Fortunately, you can't just go spraying DNA on things a lot and then suddenly have them become genetically transformed, because DNA is basically everywhere. Agrobacterium is a bacteria that is a specialized bacteria that knows how to transform things. Without getting into a lot of the details, it actually carries a piece of DNA called the transfer DNA, tDNA, and it has a whole host of genes that all support to figure out how to get that inside the cell and actually insert that into the plant randomly. That's what Agrobacterium does, and if you've actually seen a tree with a big tumor inside it, sometimes those are actually called by Agrobacterium. It actually codes for genes to cause the formation of free food and a big tumor to live in, so a house as well. Even if you accomplish that, if a tree outside has a tumor on it and it's been genetically transformed with agro, and the next generation of trees doesn't have the agro DNA in it anymore, because it's up into the seeds of the tree. You have to figure out how to make that specific cell that has the DNA in it regenerate into the actual plant itself. And again, this is a process called somatic embryogenesis. This is actually somatic embryos of the chocolate tree, for example, some work we did there. This is a pine tree somatic embryo, and then if given the right nurturing environment, it's kind of like intensive care, they'll actually go into full-blown embryos. These are somatic clones now. And so from that one, it'll divide 1,000 times. You'll have 1,000 clone trees, and then you can get those to regenerate a clone. In this case, this is a pine tree. And this is work we did almost 20 years ago with Weyerhaeuser, and it's a way in which you can actually take a tree which is a superior, taller, straighter, faster growing, and then end up with one that will ultimately give rise to that. So this is called cloning, but of course there is no genetic engineering involved in this process. It doesn't have to be. If you put the gene in, then you do it, then you've actually created a genetically modified plant. If you don't, you just simply cloned it from what it was before. So now, there turns out that there's a special case out there which meant that you can point to that does actually allow for genetic transformation by spraying. Okay, and so you can kind of imagine then that that lends itself to the scenario here of, whoa, why don't you just spray things? Arabidopsis is the model plant. It's a very small plant, little tiny thing that grows in the cracks of the sidewalks. And its genome was really small, so back when it used to be really hard and expensive to do genome sequencing, it was the first one for the plants. So we would have a model plant to know that that would be the basis of that. It turns out that if you actually put agrobacterium on that and you actually spray it into the flower stalk, called sleep, and take those seeds and put them onto plates, you can actually find the seeds where the transgenic ones will live because there'll be a selectable marker on there, and the non-transgenic die. This is super useful for research purposes because it gives you the ability to quickly introduce genes and go straight to the next generation. Okay? So you don't have to do that transformation, go undergo somatic embryogenesis, grow the thing up, get the seeds, and then test them. You spray it, and then the seeds go right directly to the next generation, the F1 generation. Now, you look at that and say, well, that sounds like it's easy. Fortunately, it's not. Well, fortunately, it's a very convenient thing for molecular biology, but it's more or less an anomaly. This doesn't normally happen. You can kind of imagine that if I'm a plant, I don't particularly want easily to be genetically modified into my next generation. And there's a lot of layers of protection against DNA being introduced into the seeds because I want to preserve that. And if it was, remember, there's no insects and there's no virus. Okay? So if this was the way that we were thinking about worrying about a bioweapon in which you could do genetic modification and spraying, and this would actually work in our crop plants, which it doesn't, okay? Then we wouldn't have to be worried about this DARPA program because spraying something like agrobacterium could be done without any of the technology development we're talking about here. Okay? It just is not that particular scenario. So now, agrobacterium has a lot of tricks that people have figured out to it. It is used a lot in what's referred to as the transient expression that I explained. And so in the transient expression, you literally take the bacteria and you squish it through the stomata. If you actually squish it through the stomata and it's in the air spaces in here, it'll do what it does normally. It'll actually take the DNA, and it'll push it into inside the plant cell, and it'll undergo transient expression. So this is a syringe here that you see, and that's, again, it's a model plant. Nicotiana benthamiana, this thing works really good for this. But it's used for this because it's easy to do something. If you want to actually squeeze the DNA in there and have it expressed, you can harvest the leaves 5 days later and see what the function is of that gene and not have to wait for 5 months for a transgenic plant. So it's a very common technique that's actually used in association with plant biotechnology. Agrobacterium is it, and it kind of looks like that whole thing of, well, why don't you just spray it? But in the process of doing that, you don't get transgenic seeds from this process. You just simply get a very quick transient expression that you can test 5 days later instead of 5 months later for the purpose of doing genetic transformations. Okay. So at any rate, that's the end of the sort of the technical background, so you can kind of get to the challenges here of what they are saying as far as scenario here. First of all, Cas9 is big. Not too surprisingly, it has to accommodate the ability to latch onto the DNA with cutting functions of the DNA, accommodate that whole guide sequencing RNA and all the other jazz and so forth. So Cas9 is pretty big. The one that was originally used, you can see, is over 4,000 base pairs. Now, they found smaller versions of it, okay, and this was actually in Nature Communications. This was they got the whole way down to something that was just under 3,000 base pairs, okay. The virus, this is huge, by the way. This is a very big enzyme, okay? And remember, we're having to deliver it with a virus. The virus that we're working with, it has 2 parts, but the part that actually has the replication functions and so forth is less than 3,000 base pairs. So it would require replacing all of the DNA of the virus in order to try to deliver the actual Cas9 into that system. And of course, that means that without that, okay, so if you try to make it bigger, it doesn't code. Okay, so this structure, you can see in some electron micrographs of that, is very, very specific. If you mess with the size of the DNA, the genome of this thing, it won't encapsulate, it won't function, it won't transmit through insects and so forth. So you can't just say, oh, let's just slap in some extra stuff and make it work. There are different forms of that, but I'll talk about in a minute. Keep in mind that we still need the guide RNA, a promoter, a payload, and all that stuff. And we haven't even had the Cas9 there. Okay, so this is, again, getting to the issues of what we're doing specifically in our project. Now, the other thing is that viruses are notorious for getting rid of DNA that they don't like. You can kind of imagine that a virus is a really intense collection of functions to be a virus. Okay? If you try to put something in there that is not a function that helps the virus replicate, make thousands of copies of itself, get to the next generation, it just gets purged. And so there is a very rapid issue here, and they become particularly unstable. I mention here because there are viruses that are plenty big to put these in. They're much bigger viruses, but they're almost all RNA-based viruses. And the thing about RNA-based viruses is that RNA is less stable than DNA, and they're even more prone to actually mutation and loss of the genes that you would actually put in there as well. And so, at any rate, that gives you sort of the idea of some of the challenges that we have associated with making it work. Now, I wanted to run this video if I could. Okay, they're really worried about transparency, whether or not people are explaining what it is they're actually doing. Okay. What I explained there is a lot more than what most faculty would ever say about what they're doing, not because they're trying to be secret, it's because they don't want someone else to steal their ideas before they get a chance to actually publish it. Okay? And so the intention here, this is actually, it's a public one, video now on the web that actually shows the method, how this thing would actually work. Now, what I wanted to do here then is get to the next piece of this, and that is, how is it different from what science presumed? Okay. They looked at a description of what DARPA thought might be the way to do this technology without asking anyone what they were actually doing. Okay. And then they waited for a year and they read what they should have done and then assumed that that's what had been done, and they wrote an article. Okay. First, you'll notice we don't use CRISPR. It's sort of the most obvious, right? Second here, it's not a possibility. Our system isn't big enough to actually accommodate CRISPR in the actual system itself. Second is the focus was on biocontainment. The virus actually needs the transgenic plant that has the missing pieces it doesn't have in order to actually function. And so that gives rise then to another means by which biocontainment, there's a lot of other layers of biocontainment we don't have time to get into. We're actually not even delivering genes, we're doing functional regulatory elements. We're taking microRNAs, things like that, and the intention here is to actually use the natural defense of the plant to actually, through human intervention in dynamics and timing. Let's very different than scenario that is done in the other one. So with that being the case, then I have just two slides here before just the Q&A here. The preferred comments and lessons. Okay, one of the news outlets actually put the get your facts straight was in the article, because there were some problems even with the science article itself, which is kind of hard to get facts if you actually don't ask for them. Okay. You'll notice here that the first one is the actual call for proposals. Okay, that was just what they said. The public document says we would like to protect crops. Here it is. Here's the details, so on and so forth. That's their number one reference. The next are the announcements of funding. Who got the money? Okay. And if you take a look, these are web pages. And half of the 15 references are web pages. I noted in here for this class that if you handed me a technical paper with half of these, you might get a 5 out of 10. Not exactly rigorous reporting. Throwing in here, okay, I have a few, and this is the Q&A part of where I have it, where I have a series of quotes, which I'll throw up there, which are from the actual paper, to help prompt if anyone has any questions. They're envisioning that if you were to spray it on the plant, nothing would happen. So, there are viruses that are easily transmitted, like tobacco mosaic virus. It's really easy to take tobacco. In fact, if you work in a tobacco farm, you can't smoke. You just touching a cigarette will be able to transmit the TMV. But the viruses here that we're talking about are actually as hard, because not only do you have to get it into the vascular system, you have to get it into the phloem. If it doesn't make it into the phloem, it's not going to actually work. And spraying that is not going to do anything. It's not going to work. Other questions? Take a look at the ones that I had here. Of course, the first one that I threw up there. There's some interesting aspects up here. I thought, in particular, start in the bottom here. Well, I like that one because it's a way to get some really things. I'm not really saying this, but you just did. Okay, and so forth. I have thought some of the aspects here that I thought were quite significant. If you take a look, I mean, two of the three are actually working on corn. Here's the double-edged sword. We could be protecting the weed, Arabidopsis, okay, or we could be protecting corn. The differences of the viruses and the method for a different type, a different crop, are going to be different. If I develop a technology to protect weeds, again, I would have to start over again to try to figure out what I might do or couldn't do if I was trying to protect something that was not. So, yeah, it's true. It is, quote unquote, more impactful and I suppose more dangerous to work with a crop that's actually important. Okay? Your other alternative is to work with something that nobody cares about. Okay? And then that's something that has to be balanced with the biosafety aspect. And that's why this is not going to the field, it's all under full containment, so on and so forth. That's the intention of the system, is to do a feasibility type study that's there. The other aspect of this that I thought is an interesting, and that is that even if they stop funding it, it's like you open Pandora's box. The fact of the matter is the technology is already there. Okay? And yeah, it's true. You can say that, oh, well, DARPA did this, and therefore they shined a light on it. Well, they shone a light on it for people who never cared. Okay, if someone really cared about the bioterrorism, they didn't need to have DARPA shine a light on it. At least that's my opinion. Okay, many of the elements and so forth are already there. So the idea that we're creating something de novo is not. It's literally decades of research that have the details that make even the feasibility, the ability to even conceptualize this possible. You go back 10 years and you would actually put this out, everyone would just laugh at you. Okay, we are getting where we can look at this thing and actually at least look at the scientific details. Part of the problem, of course, with a document like this is that it isn't really about scientific details. It's neither long enough nor did it try to find them, which is part of the problem. CRISPR, let's see, stipulated. Yes, this was relevant because it gets right to the one here. They maybe inferred that we said we weren't using CRISPR, so they had to say something. Whether or not chromosomal editing is used to achieve the stipulated easy simplifications. By the way, easy is not a word that goes very well with this project. Okay. Of the described work program could be used to generate a new bioweapon. So they just want to cover their bases here. In case I say, I'm not using CRISPR. Well, I mean, I could be using a spray bottle. Okay. If I was going to terrorize a rhabdomatosis. Okay. And so I think that recognizing the fact that we are going to be dealing with, climate change, okay, or whatever word that you want to replace there, is part of the motivation behind this. It's that basic idea that we better be prepared for this. You get a spring planting, and then, oh crap, a month later it's going to frost. Maybe I could do something about it, or I could just lose it all. That's the scenario that we see here. There are other scenario that I think is also important is we have a tendency to think of crops as annuals. You stick stuff in the field, you grow it, and then you harvest it, and then it dies. Okay? The reality is another motivation is for instance, the loss of the oranges in Florida. Okay, there's some terrible things that are going on there, killing all the orange trees in Florida that are there. I mean, it took that farmer 20 years to grow his crop of orange trees. Okay, and so how can you protect that? You say, oh, well, just go breed a disease-resistant variant of tomatoes or of oranges. Take another 10 years and then plant them, and then wait for another 20 years and be like, I'll be dead before you have a solution. I think I'll just not grow oranges anymore. Okay? That's the scenario that and there's an awful lot of things on this that we have besides trees and perennial grasses. Like for this class, we talked about doing a lot of stuff with perennials for biofuels. If something goes after those, what are you going to do about it? You have a choice. Nothing. That's one. Replant it. Wait another 5 years. I guess those are the scenarios of where you can kind of see that something like this might actually deal with a mature plant that's out there. Not, 30 years from now we'll have a GMO orange tree that won't have that particular disease problem.